Welcome back, W and gang. Today, the biology bros are going to be talking about CRISPR and gene editing. CRISPR is a revolutionary gene editing tool originally found in bacteria. It allows precise DNA modifications in living organisms. Off to my boy Owen.
So far, applications of CRISPR technologies is curing genetic diseases in animals and humans, for example, sickle cell disease and muscular dystrophy. It could also improve crop yield by making crops drought resistant and pest resistant. An example could be, wheat crop. Maybe that's a good example of a crop that could be.
Very useful for bread and bread making.
It could also enhance cancer treatments with gene-edited immune cells that could be that can target specific cancer cells and tumors.
Now, there's also further potential of gene editing in CRISPR. It can help treat genetic disorders in humans or in animals. This possibly can help make a more sustainable living for humans and make us advance further with technology and advancements. Also, we can help modify mosquitoes to eliminate malaria, which is a very dangerous disease found in Africa and South America. Lastly, we can engineer disease-resistant livestock so that we can have more food for humans and more food for animals that hunt these animals. Like cows or chickens, and for example, wolves and coyotes hunt these animals a lot. See you on the next part. Oh, and I have a question. How does CRISPR work?
Well, CRISPR works like molecular scissors for DNA. It uses guide RNA or gRNA to find and locate a specific DNA sequence in DNA. It then utilizes a Cas enzyme, CAS enzyme, for example, Cas9 or CPF1, and it cuts the DNA at the target site.
Oh, okay.
It's almost like a spliceosome and RNA, which helps, not RNA, a spliceosome and
pre-amount of RNA.
No, not exactly. It works like a spliceosome, takes apart the introns, and puts the extrons back. It works like that.
That's pre-mRNA, buddy. All right. Also, after you do the CRISPR procedure, the cell can repair itself, leading to gene knockout, disabling a gene, which will ultimately, maybe that's what the person wanted to do or not wanted to do. So there's pros and cons, both. There's also gene insertion, adding a new gene, which just you add a new gene and it possibly turns off another gene or affects another gene.
I have an idea for that, man. Maybe if someone wants to get like reptilian scales on their arm instead of human skin, they could insert the reptile gene and make their skin like that.
I like that. And lastly, there's base editing, which you just change the DNA letters, which ultimately just changes something up in the cell, and after meiosis happens, the body will start adapting to that.
So if you think about it, Matt, isn't that kind of like the substitution in RNA transcription?
Yeah. It's actually mutation, then you're mutating your body. So it's also not perfect because it can cause off-target mutations, which means that if you maybe turn on one gene that you wanted, but another gene might turn on or off, which will ultimately affect you and make you look worse or better, depending on.
This is a major issue ethically and technically with CRISPR technology. This is one of the reasons why we have not seen CRISPR applied to humans a lot, and even applied to animals and plants. We don't know its limitations yet.
Yeah. All right. What's the difference between Cas9 and CPF4, Owen?
Well, for one, the features, size, cutting style, and the guide RNA are big differences between Cas9 and CPF1. First, the size of Cas9 is much larger than the size of CPF1. The cutting style for Cas9, the technique you use is you cut both strands at the same spot, and then you splice them together after that. For CPF1, it's a staggered splicing. The sticky ends are cut off on each part of the DNA, and then they are spliced together after that.
And ultimately, I think just Cas9 is most commonly used, while CPF4 is more precise but newer. So that's why more scientists prefer the older version than the newer, because they are used to that one.
And there's less research on CPF1.
Yeah.
Okay. Right now, the biggest CRISPR therapeutics company in the biotech industry is called CRISPR Therapeutics, which was co-founded by CRISPR pioneer Emmanuel Charpentier. CRISPR Therapeutics focuses on gene editing therapies for human diseases. It focuses on humans, and it is trying to revolutionize CRISPR therapies in humans. Some of their major projects include ECE or CTX00001, which is targeting at treating sickle cell disease and beta thalassemia, which is it is currently in its late stage trials. Another project that CRISPR Therapeutics has been working on is cancer immunotherapy. They've been trying to edit immune cells in the human body to help fight cancer tumors, both malignant and benign, as best they can. And finally, they've been researching diabetes and heart disease treatments to try to reduce and eliminate those diseases from the current how common they currently are.
Add Zach, add Carter to your group too.
Ethical and societal debates around CRISPR. Human embryo editing. Should we allow it? And what are the risks of designer babies?
Should we be allowed to give babies chopped chin cooked features?
I don't think so. You should keep it how nature wanted it to be. While you're making these designer babies, there can be unintended mutations that can happen. Since CRISPR isn't 100% accurate, and it could cause harm to the baby instead of improving it. Bioweapons and misuse could be used to create harmful organisms, which you don't want because it might kill the mother in the process. Accessibility and inequality. Who gets access to CRISPR treatments? You need to pay a lot of money to get a designer baby, so as a result, it's not going to be fair to everyone.
Yeah, CRISPR treatments are very expensive, so it's a very big deal what kind of people are able to use it. Currently, only people with a lot of money, a lot of donors, and companies that have been funded through grants are able to use CRISPR. And if only the rich and elite are able to use it, it's not a very useful technology.
Yep. And lastly, there was a case study done by Naki, a Japanese scientist, in 2018, who illegally edited human embryos, sparking global outrage.
That does not sound like a very good.
Yeah, he was doing them illegally, so pretty bad.
No big booms for
Yeah, zero big booms. My partner has left, so I'm gonna have to do the next two parts. What is the future of CRISPR in science and medicine? There are more FDA-approved gene therapies expected soon, as a lot of them are in the late stage of development. There are newer CRISPR techniques, base editing, prime editing. Those will improve precision and make CRISPR more accurate to what you want it to be. Personalized medicine, tailoring treatments to individuals' DNA. This will help humans and possibly animals in the agricultural stand, as they will not be able to get diseases, and it will make people healthier and animals healthier. Agriculture, it can help crops be climate resistant, which means that crops can grow all year round, except instead of one season. Like apples, you can pick them in the fall, or wheat, you have to cut it down during the summer. But the biggest question is, will we use CRISPR for human enhancement or just disease prevention? There's a lot of controversy on this topic, as a lot of people want it for both, but some people think that human enhancement will make us advance too quickly, and it can possibly bring our world to an end. My take on CRISPR's future. CRISPR will revolutionize medicine. It will not happen overnight, but in the next 10 to 15 years, as a lot of them are in deep stages of development. Genetic disease treatments will be the first major breakthrough in CRISPR, as that's what I think, because human editing will be much more advanced, and it will probably happen in 15 to 20 years. Now, after my partner, what does he think?
I think my take on CRISPR is that it'll have major applications in the agriculture and food industries. It will make food more secure, and agriculture, the system as itself, more secure, because plants will be a lot more consistent with their growth and the genetics, and it won't need to rely on pesticides and topical treatments to the plants in order to achieve these better yields and disease resistance. It'll be ingrained in the plant's DNA, which is a lot more safe and a lot more consistent. I also think that human advancement is not on the horizon for us. I think human advancement is not. There's too many ethical concerns, and it will have to go through a lot of legal screening and processes, which will ultimately slow it down. I don't think human CRISPR editing will happen anytime soon.
Yeah, and then I think in the next 50 years, gene editing could redefine human evolution. But regulation will be key, as you cannot abuse this, and because it will slow down human advancement instead of speeding up, and ultimately lead to the depletion of human society.
Yeah, I agree, man. I think CRISPR biotechnology is a really similar field to artificial intelligence.
Yeah.
Right now, a lot of people are talking about how regulation of AI is the biggest thing we need, because right now they have a lot of new startup companies like DeepSeek and OpenAI, and those currently there's not much regulation. They have pretty much free reign over creating. They're basically creating a whole intelligence, and it's even smarter than humans. It needs to be regulated. Very similarly, CRISPR technologies, with all of its debates and ethical concerns, it will definitely need strict government regulation in order to make sure it is benefiting humanity and not being used for greed and monetary value.
Yep. And ultimately, I think the biggest challenge isn't if we can edit DNA, but how we use this power responsibly so we can help us and not abuse it and make our society.
Me personally, I can see a whole dystopian future, a book or a series, that kind of thing, where humans have taken it way too far, and we have everyone looks the same, everyone is the same, everything is the same, and that's not something we want.
Yep.
So Matt, all right, Matt. So now that we've reached the end of the video, how many big booms does CRISPR biotechnologies get?
CRISPR and biotechnologies gets 5 big booms. Boom, 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 boom. 5 big booms, guys. If you liked the video, make sure to like and subscribe. Comment what your favorite part of the video was, and see you guys in the next video.
See you in the next video, guys. Goodbye.
Subscribe to Biology Bros.
Subscribe.
Remember that.
Bio.